To understand the cell cycle, it'll be important that you draw a diagram to help with that. Draw a circle, make it big, about half a page, and then divide it into quarters. What you have is the three major stages of the cell cycle. The first is interphase, where normal cell activities take place. The second is called mitosis, where the nucleus is divided. And then third is cytokinesis, the division of the cytoplasm itself. Interphase has three smaller substages of its own, called G1, S, and G2 phase, whereas mitosis has four substages: Prophase, Metaphase, Anaphase, and Telophase. In order to continue with the rest of this video, there are two definitions that are going to be important. The first one is called a chromosome, and this is a condensed form of DNA with a centromere. That centromere holds two halves of the chromosome together called sister chromatids. Different than a chromosome is chromatin. This is DNA that's been uncondensed, so it's less associated with proteins, scaffolding proteins in particular, although it is still coiled up with those nucleosomes and histone proteins. And finally, the last one is the DNA, which you're very familiar with by now, and that's in the form of the double helix. In fact, I'd like to take the analogy of yarn. It's a good analogy because yarn is, like DNA, very thin but exceptionally long. So if the yarn itself represented the double helix, then the mass of yarn here is called chromatin, which is very useful because you can use any portion of this yarn or any portion of the DNA. However, it's not very portable. So when it's time to move the DNA, then it's going to condense in a form called a chromosome, which is far more transportable but not very usable. So when we find ourselves in interphase of the cell cycle, interphase is where normal cell activities take place. This includes things like growth or metabolism, homeostasis, all of these things that the cell would normally be doing. And in order to do that, the cell is going to need DNA in the form of chromatin. The second stage is called S phase. At some point, the cell receives some kind of signal that suggests it's time to divide. And so the DNA is replicated, so that chromatin is copied. Now I have two exact copies of that DNA. The third stage is that now that the DNA has been copied, the rest of the organelles are copied in preparation for mitosis. Later, in a phase called prophase, that chromatin condenses into chromosomes. And of course we have two halves of that chromosome because we had two original copies. So here we have two portions called sister chromatids, and the cell keeps them together with a structure called a centromere. So in short, we have a replicated chromosome which has two strands of identical DNA called sister chromatids, which are held together by a centromere. Human cells contain 46 of these chromosomes. And so the cell cycle looks like this. We have interphase with G1, S, and G2. At the end of G2, the process of mitosis begins. Mitosis is the division of the nucleus, and we begin at prophase. Looking at the two cells on the right-hand side, you'll see that the first one is an interphase cell, and the second is a prophase cell. And looking at the two cells carefully, you'll see that there are distinct differences. See if you can find these as we go through it. The first difference is that a prophase cell doesn't have a distinct nucleus, because at prophase the nuclear envelope and the nucleolus begin to break down. You'll also notice that what was chromatin in interphase has now condensed into chromosomes, and each of those chromosomes has sister chromatids held together with a centromere. And the third is that structures called centrioles have formed and begin to move to opposite sides of the cell, and between them are spindle fibers made of microtubules. Consider these like two spiders spinning a web between them as they traverse the cell. The second stage is called metaphase. Here in metaphase, the centrioles are going to travel to opposite ends of the cell and anchor with the spindle fibers to the cell membrane. Secondly, the spindle fibers that have extended from the centrioles now attach to the centromeres of each chromosome. And as they do that, they guide the chromosomes to the metaphase plate, or the equatorial plate. Whichever name is used, realize that they're simply identifying the middle of the cell. And in fact, that's what metaphase means, the middle portion. Next, we have anaphase. An anaphase begins by the centromeres of each chromosome split. And as they do so, the sister chromatids separate from each other and move to opposite sides of the cell along the spindle fibers. After, though, these chromosomes have been separated, the chromatids are now considered individual chromosomes. So ask yourself, how many chromosomes are in this cell at anaphase? The last stage of mitosis is called telophase. In telophase, we begin to see the undoing of prophase. So the first thing happens is that the nuclear envelope and the nucleolus begin to reform. Chromosomes begin to uncoil back into chromatin, and the centrioles and spindle fibers begin to disappear. And though this looks like the undoing of prophase, there is one great exception. Here we have a cell with two nuclei, and so the cell is now going to start dividing the rest of the cytoplasm. It does this by separating the cell into two halves. In animal cell, the cleavage furrow begins to form, a fiber that begins to separate the cell in half, or in plant cells, a cell plate begins to form. So we have the complete division of the nucleus, where now we have interphase complete, mitosis is complete, and now the remainder of cytokinesis must be finished, dividing the cytoplasm. And so the completion of that cleavage furrow in animal cells, or the cell plate in plant cells, will divide the rest of the cell. What are the results? Well, we should get two daughter cells with two nuclei that are exactly the same. These cells should be indistinguishable from each other on a genetic level. And secondly, these cells should be half the size of the original cell. So take some time now and respond to the 3 questions as seen on the reflection page. Make sure that you explain your answers whenever necessary.